Bailey, and let's continue going through the IB past papers questions of multiple choice related to the topic 1.2. So we got about 13 questions. Let's get exposed on how to tackle these guys. So here is the first question. What distinguishes prokaryotic cells and eukaryotic cells? So for this, you have to go back to the memories of what prokaryotic cells and, of course, eukaryotic cells are. So we got to look at the first sentence here. Prokaryotic cells have cell walls. That is true, because if you remember the bacteria, the outer wall, the outer part is made of cell wall, right? And then the eukaryotic cells have a plasma membrane. Well, that is true. Animal cells, they have a plasma membrane with a nucleus, right? This is the plasma membrane. So this is more likely to be the right answer, but always check the next ones, right? Now, what distinguishes between them? Let's say here, this one has cell structures not compartmentalized. That means when I say this word not compartmentalized, it means it does not have organelles, which is also true, right? And that means it's okay. And this one, cell structures is, which that means yes, they do have organelles. The next one here says that prokaryotic cells have the smooth endoplasmic reticulum, while the eukaryotic cells have the rough endoplasmic reticulum. No. First of all, we know that prokaryotic cells do not have any organelles, so this is out of the way. Now, the next one is prokaryotic cells have no ribosome. That's not true. If you remember, they have 70S, and while the eukaryotes have S, so that's no. So now you're stuck on these two. This is what usually students are thinking. Cell wall in prokaryotics, while eukaryotic cells have plasma membrane. All those are true, but there's a huge but. Eukaryotic cells is also a plant cell, besides an animal. And what does a plant cell have? Cell wall. So cell wall is not what makes the difference between prokaryotic cells and eukaryotic cells. So that would not be the answer. So the final answer in this all case will be B, because again, the word not compartmentalized means it has no organelles. And yes, prokaryotics do not have organelles. Let's go to the next question. What is the process shown in the image? If you remember from bacteria, right? Bacteria, this is a bacteria drawing here, and it has basically its DNA. Now, when you want to go through division, first thing you do is create an extra copy of that DNA. And then the bacteria will grow a little bit more. And then, of course, each DNA will go to one side, right? You have a DNA going here, and you have another DNA going here. And eventually, with time, you will have two bacteria split. And this is known as binary fission. So if you look at this drawing that I did and you compare to the picture that it's right here, you can see that this resembles this. So going now looking at the question, it says what process is shown. So it's a process in this image right here, right? You can see that it's bumping. So let's look at the options. The first one says it's a binary fission of prokaryotic cells. This most likely looks like an answer, but read the rest. Telophase 2 in eukaryotic cells. Not really, because it wouldn't be looking in this kind of shape. Especially this looks more like a rod. But some students will start wondering, oh, this could be. But if it does, it would have been a darker side here, and it wouldn't be all this white part here. So I would still say no. The end of mitosis in prokaryotic cells. First, just the word mitosis. You cannot use the mitosis in prokaryotic cells. The name is binary fission when it comes to prokaryotic cells. Never use the word mitosis on it. So, not this one. Cytokinesis of eukaryotic cells. Now, that looks like a cytokinesis. Yes, it looks like it's contracting like a cleavage furrow. But what it doesn't give away is that usually they are more rounded like this, like more like a circle, not like a rod, which in this case, it would be more like a bacteria. So erasing all this to give a little bit more understanding. If it was an eukaryotic, again, it would have been more round like that. But because it is more like a rod shape, that means this is a bacteria. So D is out of the way too. So the only answer that we have in this case will be binary fission of a prokaryotic cell. That's the right answer. So the answer is A. Let's go to the next one. Here says, which substance is used for structures of plants? So we're talking about which substance here is going to be used for the structure of plants. When we say structure of plants, we're talking about keeping it standing. So what does keep a cell standing? So if you have a plant, usually plant cell, the plant cell has the cell wall, right? And then it has the cell membrane. And you're gonna have many other of those going like this and going like this and making sure like a tree can be standing, right? So what do you need? You need that cell wall. You need that cell wall because the cell wall, what it does, this part here makes it stay firm. Now, what is that cell wall made of? Now, we know that in plants, cell wall is made of cellulose. So we got to look for that answer. It's not amylopectin. Cellulose right here. Collagen can cancel, starch you can cancel. So which substance here is actually structure of the plant? In this case, it's cellulose. So the answer is B. So make sure you do memorize this one because these kind of questions, sometimes there is no other way to answer unless you know by memory information. So, which structure found in eukaryotics? So now they want to know about a structure that is found in eukaryotic that has a single membrane. Now, when we say single membrane, we are basically talking here about having one layer. That means it's just one layer like this. Now, let's start by discarding. So we know that chloroplasts and mitochondria are two layers. Like a chloroplast is right here. It has the outer membrane and it has the inner membrane. So you can see that these two are what? There are two layers. So they're two membrane layers, right? And then you have those chloroplast thylakoids going like this. And mitochondria is the same thing. You can see that it has the outer layer and then it has the inner layer. This is also a two membrane. So C and D is out of the question. Then you start having the question, shoot, is it a nucleus or is it a lysosome? Because that's where students get stuck. It's like, oh, I don't know anymore. Which one would that be? Like, I have no idea. And then students start freaking out saying, shoot, I don't know what the answer will be. Well, you guys forgot that the nucleus has a nuclear envelope as well as nucleus area. So the nucleus usually is protected by a double layer too. And many students don't know about that because it has to protect that DNA. So nucleus is also a two layer. So the right answer is a lysosome. Lysosome is only made of one layer. And that's the right answer. So the answer is B. Let's move to the next one. Which of the following electron micrographs here, in this case, shows a part of palisade mesophyll cell? So just by reading this, we are talking about a plant. You should have known this from 1.2. You have to know that palisade mesophyll is a plant. Right, so it's a plant right here. Now, which slave structure controls the exchange of substance? Now, this is just to throw you off. If you read the next question here, what it's saying is which of the labeled structures, that means here A, B, C, D, and then here's the most important thing. It says controls the exchange of substances to and from the cell. That means there are things that are coming in and out of the cell. So what we have here is one side of the picture. Now, for those who don't know this, if you look at this shape right here, this whole drawing here, this is a chloroplast. And the chloroplast, you easily can see the thylakoids, the granules here, right? You can easily see that. And of course, you might have some starch grains. So this is a chloroplast, obviously. Now, then you know that it's a plant. When you think about it's a plant, we know the plant has a cell wall, which is a double layer like this. And then after this, it has the plasma membrane comes inside, right? And then you will have the chloroplast. So we know that whatever that comes in out of the substance cell has to be a plasma membrane, because that's what plasma membrane does. See, the cell wall, this part here, the cell wall, it does support and protection. It does not control what comes in and out. So the cell wall is not the answer. So the cell wall is the outermost. Since it's the outermost, that's B, cell wall. But since we know that cell wall is not the answer, so B is not the answer. The next thing, we got to look at A. What does A do? It's pointing to the chloroplast. Well, chloroplast does not control the exchanges, so it's not that answer. So now you're stuck between C and D. Now, D is all the way inside the chloroplast, so even if you don't know what it is, that D, obviously it's not the chloroplast that is controlling the exchanges or anything like that. So the right answer in this case will be C. C is the answer. So when you come to these kind of questions, when you have labels, the only thing you have to do is just circle, or of course, if it's a paper-based, you just write C. C would have been the answer for this question. Now, let's go to the next question. Which organism has the DNA located in 3 organelles? So we're talking about DNA. We're talking about this DNA here, and it has to be located in 3 organelles. So within one, we're basically saying within this one piece of organism, I don't know, can it be a plant, can it be an animal, can it be a fungi, can it be bacteria? Now this organism, they're basically saying that there is 3 parts inside of this organism that you can find DNA. So then you start questioning, where do we usually find DNA? If you remember from drawings, you can find DNA in the nucleus. You can find DNA in the mitochondria. And you can find the DNA in the chloroplast. Now, because of that, this one gives away. Chloroplast is a plant, right? So that's one thing that we have here. Plant. That means I have to look at my 4 options and see if there is a plant. Sponge is not a plant. A fern is a plant, so this might be the answer. Flatworm is not a plant, and bacteria is not a plant. So we can even cancel bacteria because we know bacteria has no nucleus, right? So even if you don't know these guys, the sponge, the fern, and flatworm, then you gotta be stuck a little bit thinking, what is a flatworm? And it's an animal, but it's an organism. But flatworms, do you ever saw a worm doing photosynthesis? No, you never saw a worm, so I can cancel this. Then the sponge. The sponge is that you basically wash your body. Did you ever see it do it photosynthesis? No, so I wouldn't take. So even if you don't know what the fern is, you can just do the guessing. But fern is actually a type of plant, and that is the answer. So the answer is B. So let's go for the next question. Which organelle is involved in generating vesicles destined for the cell membrane? If you remember from the drawing, here is the cell, and you have the nucleus, right? Let's put the nucleus here. And then you might have the rough ER here and the smooth ER, and then you might have the Golgi here, and you might have the mitochondria and whatever extra lysosomes here. So they're talking about vesicle. Vesicles is this little tiny thing that is moving inside the cell, and it might be moving out, right? Or it might be bundling in to go inside. So they're talking which of these organelles? The rough. Is it the rough? Is it the smooth? Is it the Golgi? Is it the mitochondria? Is it the lysosome? What of these guys are actually involved in generating vesicles? Now, here is where there is more detail. It says destined. That means his final destination is what? It's the cell membrane. So let's get it clear here that whenever there is a vesicle popping out of the rough ER, that vesicle is always going to go to the Golgi. And whatever vesicle that is popping from the smooth ER is always going to the Golgi. So you already can cancel the smooth and you can already cancel the rough. These two are not sending to where? They are saying to here, it wants to be sent to the plasma membrane, or should I say the cell membrane. So those two are out. Now you have lysosome and Golgi. Now lysosome is There is no thing, like lysosome is so small, it cannot just pop out of himself another vesicle. So that's already canceled. Now we know for sure that the Golgi apparatus is the one that pops its vesicle, and this vesicle moves in the direction of the plasma membrane in order to send its antennas, its glycoproteins and stuff like this. So the answer is A. Let's go to the next question here. It says the image shows an electron micrograph of mesophyll cells. Again, mesophyll. Students freak out. What is mesophyll? If you did the studies, you know the mesophyll is a plant cell. So mesophyll, palisades, these are all plant cells. Now, if you look at this picture, you will see that these little black things here, let me put it with the highlighter because it makes it much easier to see. These little black things are chloroplasts, all of them. Oops, not this one. Can I go backward? Yes. Oh, and then here, all of these are chloroplasts. Now, what is this blue, this white thing that is all here? This is actually a large vacuole. And you have another large vacuole here, right? And then you can see from this that this is a plant. Now, that shows a micrograph of a plant cell. The next thing they're gonna look is the question. It says, what is the name of the structure labeled X? Oh, since it's already giving away, right? From my drawing, it's those black ones. So it's these guys, these black dots. As I mentioned, that's a chloroplast. It cannot be a nucleus, but the nucleus will be way bigger. It wouldn't be a mitochondria, it wouldn't be cytoplasm, because the cytoplasm will be the whole thing. So this is a question of you being exposed many times to the micrographs of plants. So if you don't know, go ahead, get back out there, look at BioNinja, look at your book, look at Google Photos, and look for plant cells in micrographs, and look at the pictures. You will notice that this is definitely a chloroplast. So the answer for this one is D. Now, next one is what evidence? We're looking at an evidence, okay, from an image of paramecium. And the image of paramecium indicates whether it's a prokaryotic or eukaryotic. This goes back again to topic 1.1, but we also are talking about 1.2, which is basically the differences between eukaryotics and prokaryotics. In this case, we know the paramecium, and this we know it because you have studied the paramecium. Mecium here is actually an eukaryotic. And the reason why it's eukaryotic, because even though it's a one cell, it has its own nucleus. So we already canceled this one because it has a nucleus, and it cannot say it's a prokaryotic because prokaryotics don't have nucleus. So B and D is gone. Next, we're going to talk about the A and C. Compartments means organelles in this cell. It's true. If you have organelles, you are an eukaryotic. But does paramecium have organelles? Yes, they do. They have mitochondria. They also have some Golgis here and Raphiar. But just to make clear, let's look at C. C says it lacks a cell wall, indicates the cell is eukaryotic. As said before, plants, right? Plants do have cell wall, and plants are eukaryotic too. So this sentence makes no sense. Like it says, if you don't have a cell wall, that means you're prokaryotic. Well, guess what? Plants do have cell wall, and they are eukaryotics. So C is also off the table. So the only thing you left is with A. So paramecium does have compartments, and again, the word here, compartment, means organelles. So it is, our answer is A. Now, this one, the electron micrograph shows parts of the cell, so you can see this. Which organelle is the site of aerobic respiration? Now, students start to freak out and look at this picture and says, holy crap, how the hell will I know what this is? The first thing I will tell you is repetition, repetition, repetition. What I mean by repetition, when I talk about repetition, it means don't just look once or twice in micrographs of plants and animals' cells, or even bacteria. Go and analyze as many as you can. See as many pictures, because when you see, the more you see, the more you start to be able to identify all the things there is. So in here, we can see in this cell that there has to be some kind of organelle that does the whole aerobic respiration. So first, learn the function. What is aerobic respiration? Aerobic respiration is the same thing as seen as cell respiration. And cell respiration, it means you basically are dealing with energy, metabolism. And who does the metabolism energy? Mitochondria. So what are you looking for here? You're looking for mitochondria. And what mitochondria usually looks in a drawing, it will look like this with this little thing. So you see more an elliptical shape, right? So if you look at all these possibilities that we have here, then we gotta look at A. A, it can barely see what it looks like. So we can skip A for now. Just look at this. Look at B. You can discard B. Why? Because it's not a rod shape. It's more like a circle. So it's definitely not B. So I already canceled one. Made my life easy. Now look at C. C, on the other hand, let's highlight it to make it more easy to see it. It does look more like a rod shape. So there is a possibility that C would have been one of the answers, but we don't know. Let's look at the other one. Let's look at D. D is this area here. And this has not even getting close to look like a mitochondria, right? It doesn't look like a rod shape like this guy. So I will also cancel D. Now going back, now we are stuck between A and C. Let me erase this part here so you can see. So you have A and C. Now the A is a little bit unclear on the drawing. And yes, IV can be such a pain on this case. But look at this. This is just this part. It still looks like a circle. And usually mitochondria is a little bit more bigger than this. So mitochondria in this case, I would say that it's C. So the answer is C. Now, how do I know this? Because I have seen so many times the electron micrographs of organelles, of cells. This is the best way they can do it. But remember, it has to be more like a rod shape and not like a circle when you talk about mitochondria. How did I know it was mitochondria? Because I know the mitochondria does the aerobic respiration, which is basically producing energy. Okay? So the answer is C. Now let's look at this one. So it's another micrograph, and I'm going to tell you from now, there will be a micrograph. There's always a micrograph in any of these tests. So the image here shows a phagocytic white blood cells. That's the immune system. So if you haven't got to that topic of immune system, you're going to be freaking out. But don't worry, we're not there yet. So it's a white blood cell seen in the electron microscope. Now, if it's a phagocytic white blood cells, most likely in this case, it would have been like a blood, right? So if it's a blood, it must be an animal. Now, to continue reading the questions, which feature can be found both within the cell and in the photosynthetic bacterium? So this cell here, because it said blood, I already know that it's an animal. So they're saying in this animal plus a photosynthetic bacterium, that means in the bacteria. But this bacteria is photosynthetic, that means this bacteria does photosynthesis. And if it does photosynthesis, it means that this bacteria has what? Chloroplast. But animals don't have chloroplast, it's just plants, right? So it's definitely not going to be chloroplast. So I can cancel the first question. See, because again, animals do not do photosynthesis. But we want to find something that both of them have, animals and bacteria. Multiple nuclei. We know the bacteria has no nucleus. And because of that, we can cancel this. So now we have two things left. We have basically 70S ribosome and a lysosome. These are two things that we can be working on. Between these two, what we have here is trying to search, right, the answer. Now, some of them have ribosomes, right? Animals have ribosomes, and some of the bacteria also have the ribosomes, right? Now, we're trying to find something that it's very much feature between the two of them. So we know both of them have ribosomes, but usually animals have 80S and bacteria have 70S. So here I would say no, but then here is where I tell students they fall in the trick of already saying that it's D. Well, guess what? There's no such thing as lysosomes in bacteria. Bacteria do not have lysosomes. So that throws off D. So then you're like, but miss, I learned that animals, eukaryotics have 80S and prokaryotics have 70S. Well, but there's always exceptions. That's why you gotta read the 4 options. So the answer here is C. It has ribosomes. Okay? Because just because eukaryotics is mostly 80S, it does not mean that there might not be another animal that has a 70S. It can have 70 and 80s, but it's most likely that they will have 80s, but there's always exceptions. So be careful, read all the 4 options. Some students, I see this in my students sometimes, because they think 7D has to do with just prokaryotics, they go and circle lysosome completely. But guess what? Bacteria does not have lysosome. Next question. What function is performed by the part of the cell shown in the electron micrograph? So this again, repetition, repetition, repetition. Like I look at this, I already know what it is. And it's not because I'm a bio teacher, it's because I have seen these micrographs every year so many times that it becomes so obvious. I know that from this picture that if I do a drawing like this, what is this? This is a rough ER with the little dots, which are the ribosomes. That's exactly what it is. You can see these little dots here. All these little dots is the ribosome of a rough ER. Now, let's continue with the question. The question says, what function? So what does this rough ER does? Is performed by the cell. So look at this. Locomotion. Locomotion means movement. If it's inside the cell, how does it move? No, it has to be on the outside of the cell, so it's a no. Synthesis of protein, this more likely. Why? Because we have ribosomes, and we know that ribosomes, what do? They produce protein. So you have to remember all the organelles and their functions. Movements of chromosome. First, if this was the nucleus, a nucleus would have been looking like this, like a very large circle, black. You don't see any of that, so it has nothing to do with DNA chromosomes. It's out. Breakdown of cellular organelles. If it was like this, it would have been a mitochondria like this, and you have it like this. So it will not. Now, some students will say, well, miss, but if you really look at the mitochondria has these lines like this. Yes, they do, but you wouldn't see so many black dotted lines like that. And this is not breaking down. It's mitochondria is actually making energy. This is breaking down cellular organelles. That means it's when the mitochondria is not working anymore, you destroy mitochondria. Or when a chloroplast is not working, you destroy. Who destroys? Who recycles? Lysosome. And lysosome is what? Is a circle. So it would have to be, in this case, a circle like this. And this is not even close to circle. So that you can automatically know that this has to do with the synthesis of protein. So the answer is B. All right, the next question here. It says the image shows an electron micrograph, and you can see, and we're going through so many electron micrographs of a virus. So you already gave you the answer that it's a virus. Okay. It's electron microvirus articles known to infect a bacteria. So it's a virus that is affecting a bacteria known as Vibrio parahemolyticus, I think. God knows what those names. Which is associated with gastroenteritis. Now you see these big names and you start to say, holy phew, my God, how am I supposed to do this? Don't worry, usually I put those big names for no reason. So keep reading the question. So it's associated with gastroenteritis. Gastroenteritis, this is actually means it has to do with your small intestine. So do not freak out. That doesn't throw you off. You don't even have to know that it was about small intestine. Keep reading. So, which is associated with gastroenteritis wound infections and septicemia in humans and animals. So basically we're saying that it's a virus that causes infections. That's it. From this whole thing, just say it's a virus. It's a picture of a virus that causes infection. Who cares about the rest? Now, look at the bottom. The question says, what does a virus have in common with a living cell. So some students will go and freak out because they see so much, big names in this thing. And then the question comes and says, hey, what is it that virus have in common with living cell? Duh, like you went through this whole panicking. So please read the entire question carefully. So virus, usually it is made of a protein. So this is a protein coat. And it has a protein legs. And inside the virus, there will be usually just genetic information, which it can be DNA or RNA. So it can be RNA here or DNA. That's it. That's what proteins are made. They're just protein, DNA. The living cell, we have the nucleus, mitochondria, chloroplast, lysosome, raphiar. So they want to find something that is common between them. So when you see things that are common between them, first, I just told you that it's only made of protein and genetic material. So you need to find, does a living cell have protein or genetic? Yes, it has both. So you have to find in the 4 options something for proteins or for something for genetic material. So ribosome, it does not have ribosome virus, so it cannot be this one. Genetic bacteria, more likely. A reproduction by binary fission. See, virus cannot reproduce. That's why virus attacks the bacteria, so the bacteria can do the reproduction. So this is out. And anaerobic respiration. Viruses don't even have mitochondria. They just have genetic material, so it's done. So the answer can only be B. That's the right answer. Because again, virus only has a protein coat and it has a genetic material. Now, many students think, oh, if virus has a protein, that means it has a ribosome. No, it doesn't. It just has the protein. Well, how does he do this? Exactly, how does he do this? He needs a bacteria. A virus needs to attack a bacteria or an animal cell or plant cell in order to use their metabolism. That means use their RAFIR, their ribosome, to make their own protein. That's what it means. So with that, thank you. This is finishing the topic 1.2 multiple choice. So you can see there's a lot of micrographs. Make sure you go and look at a lot of those pictures and start to identify, and you'll be fine with 1.2.